two keynote speakers this morning talking about the development of EEG biomarkers to assist treatment development in Fragile X syndrome. Please join me in welcoming Devin Binder and Craig Erickson. So, Dr. Erickson is an associate professor of pediatrics and psychology at Cincinnati Children's Hospital Medical Center. He founded a Fragile X clinic initially in Indianapolis and then moved the program to Cincinnati. He has a large Fragile X clinic, hopefully many of you saw that last night, and sees over 200 patients. He has been involved in clinical trials for translation of targeted treatments to patients with Fragile X syndrome and autism spectrum disorders for over 12 years. He has done extensive work on biomarkers and outcome measures for use in these clinical trials. He is currently serving as the chair of the Clinical Trials Committee of the Fragile X Clinical and Research Consortium. Dr. Craig Erickson is, whoops, that's a repeat, sorry. Dr. Binder is originally from the Bay Area. Yay. Yes. I am. He went to Harvard University as an undergraduate where he studied biology, anthropology, and neuroscience. He was awarded the Hoops Prize at Harvard for his summa cum laude senior honors thesis, Serotonin and Behavioral State. Deciding to pursue both neuroscience and clinical medicine, he enrolled in the MD PhD program at Duke University. At Duke, he graduated first in his medical school class and contributed to epilepsy neuroscience with his PhD dissertation, The Functional Role of Neurotrophins in Kindling Model of Epilepsy. Subsequently, Dr. Binder completed a one-year internship in general surgery at the University of California, San Francisco, and a 6-year residency in neurological surgery at UCSF. He did a one-year fellowship in the laboratory of Dr. Alan Verkman, leader in the field of aquaporin biology. I'll have to get a description of that. Following residency, Dr. Binder was awarded the Van Wagenen Neurosurgical Fellowship for 1 year of neuroscience and neurosurgery at the University of Bonn. There he did another fellowship in the laboratory with Dr. Christian Steinhauser, director of Institute for Cellular Neuroscience at the University of Bonn. Following a 3-year stint at the University of California, Irvine in the departments of neurological surgery and anatomy and neurobiology, Dr. Binder joined the Division of Biomedical Sciences at the University of California, Riverside. He is a dedicated teacher, having won awards for teaching at Harvard, Duke, UCSF, and UCI. He welcomes all those interested to apply for the laboratory for more information. So can I just say that I am so impressed by how many brilliant people are working in the field of Fragile X. I mean, the conversations that we've been having are awesome. That gives me so much hope. So welcome, both of you.
So we're going to organize our talks, which are really linked, by discussing human Fragile X work initially. I'm going to do that. And then Devin's going to talk about mouse studies. And I think really what our common theme is doing as synchronized and linked experiments as we can in mouse and human study to really sort of bridge the gap in treatment development. So I'm really honored to be able to bring basic science and human science together in a really linked manner. So first of all, I'm going to talk about single dose drug studies in human Fragile X syndrome. So can we give one dose of a medication, see what effect it's having on the brain, and then down the line use that as a predictor of what medicine may be best for an individual? So really, this is not just my own work. Lauren Schmidt, our postdoc psychologist, has been really involved in the presentation and analysis. Ernie Petapati has developed a resting EEG data pipeline that's been quite amazing in recent months. Lauren Etheridge, who's here from the University of Oklahoma, is really the world's leader in getting EEG analysis and setup synced up in Fragile X syndrome trials and in other studies. And then John Sweeney is really our scientist that helped conceptualize the original experiments and allowed us to bring them to our Cincinnati center. So first, I have my conflicts of interest where we, you know, in research, funding is hard and we get funding where we can. So today's work is really supported by the National Institutes of Health. So if you like what you see, these are your tax dollars at work. So I think we also work with industry in many different ways in new studies, and we're supported by Children's Hospital and many philanthropic groups. So next, I want to really thank and show appreciation for the many families impacted by Fragile X syndrome in our area. And we've really worked a tireless pursuit in recent years to bring this meeting here so families could attend that otherwise are not attending. And I received dozens and dozens of photos from families. And this is really just a representation of why we do the work we do, why we strive to improve care and outcomes. And I'll continue to show photos through all of my talks because, you know, it's going to take probably all 10 talks to cover all the photos, which are really the most important thing because this grounds us in why we really do what we do. So first, an introduction to the concept, building a solid bridge from mouse to human and back. And we really have back and forth in this research pursuit. So that's the John Roebling suspension bridge, which, you know, your Cincinnati history would tell you that this was Roebling's experiment before the Brooklyn Bridge. So you start small in Cincinnati and then you move to the big time in New York City. And you know, I hope we can build as solid bridge as this one. It's not going anywhere. It vibrates a little bit, but it'll never fall. So, so the real question is, how can we utilize EEG or brain electrical activity studies across mouse and human work to assess single drug dose response and be able to predict then how a particular medicine may be matched best to an individual? And this is really bridging work that's different than what we have done in many other studies. And I have a whole list there: audiogenic seizure, novel object recognition, how mice bury marbles, how brain protein expression may change, and many other tasks. And the big difference, we believe, between the work that we're going to talk about today and what others, including ourselves, have done is that we're going after features that we believe we can directly model in human study and in animal study by studying brain electrical activity using the exact same paradigms. So we think that will build a more solid bridge. So if we see a positive response in a mouse, it'll better extrapolate to human beings than what we've done prior. And it's going to require matching drugs and dosing very precisely and matching up exactly how we do the EEG studies in mouse and in humans with great precision. And what's the overall goal? The goal is personalized medicine in Fragile X syndrome. So the right drug treatment in the right patient at the right stage of development. And I think, you know, when I entered this field about 13 years ago, I came out of the autism field, and we thought this was a single gene disorder where individuals were quite similar, and where we thought it'd be a lot easier to enter drug discovery, and that we could find one agent for a single gene disorder more easily than we could in autism, which probably has thousands of genetic contributors. But it's proven quite different. And I think anyone that is in a family or otherwise knows someone impacted by Fragile X appreciates that variation, but it took hard-headed scientists probably a decade to get there. And I think now we realize that individual differences are as important as overall similarities. And we're really appreciating that now and trying to match treatments to individuals. By using EEG and studying brain electrical activity, we think we can understand at a brain level what is going on with a treatment response, which allows you to get around placebo response, which is the concept of if you begin a treatment, a clinician, a parent, a caregiver, we inherently, whether we try to tell ourselves not to, we want an individual to improve, right? And if you do an EEG test, you can't fake the EEG, you can't make it up, and there's no placebo response, essentially. So as we personalize medicine, I think we have to move beyond solely relying on, I think someone's better, even though we know they may be. I mean, I've been doing trials for well over, almost 15 years, and, you could see in clinic who was noticeably better in a trial, but you probably had another 30 or 40% of folks that we felt were better, that maybe the changes were minimal and we may have inflated them with a good-natured desire for improvement. And that's how placebo response has plagued our work, and we're really moving beyond that with what we're doing. So this is our overall study design I'm going to discuss. And you see that we have 4 visits, each spread 2 weeks apart. Each visit is a long day where before taking a dose of a medication or placebo, individuals do a full EEG battery and other testing. Then they wait 6 hours and they repeat all of that testing. So the question is, what's the difference in the 6-hour period? It's taking that medication. So we need to see, and we're trying to evaluate early on, immediately, as a drug enters the system, is it having potentially a positive impact on the brain that would then be a signal that this may be a better pick for someone than just having everybody try one drug and then trying to sort out who's who. So we have washout periods in between to reestablish a baseline, and we have this long, 8+ hour day. I get asked a lot about what about the burden on individuals? We do pretty well getting folks through the day. We take a lot of breaks, people visit the cafeteria, they have Skyline chili, they get a piece of pizza, and that's an issue that we're going to talk about that impacts drug levels. And then they come back and do further testing. But we've done a really good job, and hopefully if you toured the clinic, it's a pretty hospitable place. And so I think that part has worked out well. So now I'm going to run through the drugs under study in this design. So the first is a camprosate, which I've studied since when my son was a baby. I always remember I was quite sleep deprived and came up with the idea to treat individuals with a camprosate. So it's like creativity through massive tiredness right after he was born. And he's here, and he will definitely give you a water bottle, a pen, and tell you all about Cincinnati sports at our table if you're interested. So one interesting feature with a camprosate is the manner in which it gets in your system is quite different if you've been fasting or you've eaten. And this is going to come up at the end of the talk. So if you remember one thing about it, remember that difference, because that's become a confounder and a challenge for us. It's a drug that affects glutamate and GABA in the brain. We've done a lot of early work with it. We've done mouse work with various assays that have looked at protein concentration, anxiety, and locomotor behaviors. It's a generally well-tolerated medicine that is, though, a challenge to swallow, as I mentioned last night, because it's quite large. So next we have Lovastatin, which is FDA approved to treat high cholesterol or hyperlipidemia. It has been studied in several Fragile X studies and animal and human work. Again, it has good tolerability and it enters the system quite quickly. And then Minocycline, which I think folks have heard a lot about. It's FDA approved to treat acne. It is a matrix metalloproteinase 9, MMP9 inhibitor, extensive Fragile X experience in human study and in animal models, and really has been a targeted treatment under development in Fragile X for many years, but I think we still, like with the other agents, don't have an understanding of who may best respond to this medication. So first of all, what is EEG? So first of all, it stands for electroencephalography. And I think what folks may first think about it is it's what you use to diagnose seizures, or if there's a question of epilepsy. We use it to really study electrical activity produced by brain cells when they communicate, when they're activated, when they have connectivity, when they're doing something. We use sensors that sit on the head. It doesn't require gel. It doesn't require sort of drilling them in. It's worn like a large net, essentially almost like a swim cap. And what can it do? It can measure the brain in action. It's non-invasive. The sensors sit there. It can be done safely at any age, even down to infancy. It's more tolerable than other things that you can do to measure the brain, such as magnetic encephalography or MEG, or some of our folks, if they have a lot of anxiety or behavioral concerns, may have trouble in MRI machines. We're able to get pretty good data in a wide selection of individuals with Fragile X, and I'll speak a little bit about that. It can also be done in mouse models to study biology in very direct ways, similar to what we do in humans. And I think that's a real strength of the modality. So what tests do we utilize with EEG? First of all, we study resting state, which is what is your brain activity like at rest? And we have individuals watch a video without sound. We do something called an auditory chirp, and I'm going to have an example of this shortly, but it's how does your brain respond to changing sound frequencies? In other words, can your brain tune in or sort of match the frequency that it's hearing? And then finally, we study auditory habituation, and this is the idea of how does your brain respond to the exact same repeated sound? Can you tune out something appropriately when you keep hearing it, or is your brain on the fourth beep responding exactly the same as the first beep? And I think this really gets to some of the sound and other hypersensitivities that individuals with Fragile X may exhibit. So this is an example of resting state, an individual in our clinic. Essentially, the cap is on, they're watching a video without sound, and we're getting several minutes of a sample. So previous findings, and this is where it starts to look really technical and it gets very colorful. So I want to get this to be as easy to digest as possible. So what we see in Fragile X outside of drug trials at baseline in individuals is what's called enhanced gamma and reduced alpha. And I'm going to speak to what these really mean. And the arrows indicate what occurs. So the blue colors there in the middle show reduction in Fragile X and alpha. So it's important to talk about, and we'll do that, what does this mean? And then the red, in particular on the sides of the brain, which we call temporal cortex, which is involved with a lot of sound and hearing processing, is increased in Fragile X syndrome. So what does resting state power mean, and what are all these bands? We talk about delta, theta, alpha, beta, gamma. It's not sororities and fraternities here. These are increasing frequencies. And there at the top right, you can see, so lower frequencies, 0.5, 1, 4, 10, up into the gamma range, which is 30, 40, 50, 60. The sound or the frequency at which it's being presented is increased, much closer together. Beep, 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 beep versus beep, beep, beep. That is what is increasing as we do these analyses from delta up to gamma. And the illustrative example there on the bottom shows how when we measure this, we can see how much power, how strong the signal is with the varying frequencies. So if you have a high gamma peak, you're having a majority of very high frequency brain activity, high frequency activity versus an alpha peak, which is lower frequency. And when we present sounds at different frequencies, the question is, can the brain match that with its own response? And, the next question, I had, and this has really taken some time to think through is, what do these really mean in the human being, this low alpha? Well, it's thought that low alpha is really involved in higher level cognitive functioning, and reductions in low alpha may be consistent with those deficits. The excessive gamma is interesting, and what I have on the right, in the A figure, and on the left side on the right part of the slide, is really representative of a neuron or a brain cell in Fragile X syndrome with a lot of little projections, too many projections, creating too much noise. It's over-connectivity. It's too much development. There's excessive protein. There are too many sprouts. Versus B on the far right, which is a brain cell from someone without Fragile X, where you have a few connections that are appropriate. So the thought is that the excessive gamma really represents too much noise. The brain is connecting all over the place and not laying down the appropriate connections that it should. And this gamma finding is pretty consistent across animal and human study. So this is the auditory chirp. And we're going to listen to it in a second. But to describe it, it's can your brain match the pitch, essentially? Can your brain waves oscillate at the same frequency as high-frequency sounds that you're hearing? In other words, can your brain sync up to what it's exposed to? And the Fragile X brain generally cannot match the frequency appropriately compared to individuals without Fragile X. So on the left, the sort of diagonal line represents that matching and that attunement of what's going on, where you see that red band showing that the brain of someone without Fragile X is able to really oscillate or signal at the same rate as what it's hearing. In Fragile X syndrome in the middle, you see that that link, that connection is much less. And then on the far right, we see the difference in blue showing the reduction in Fragile X. And now we're going to play the chirp. So really what the chirp is it's changing frequencies. It's moving to higher frequency over 2 seconds. So can we cue up the chirp a couple of times? So we're trying, but you know, getting video and audio to work in talks is equivalent to potentially landing on Mars sometimes. So I know we can do it though, because here's the deal. So, you know, I was never a Boy Scout, but can you play the chirp there? I can't hear it. Can people hear that? See, look, so like, you just sometimes you're self-sufficient. I can hear it. I don't know if you can hear it, but there you go, okay? So basically, that is an ever-increasing frequency of sound over a couple of seconds. And the question is, can your brain essentially modulate its electrical activity, its oscillations at the same rate? And in Fragile X, we do not see individuals being able to do that. The final EEG assay and study that we do, and this is the one that I describe on the back of a sheet of paper for those of you that have been with me in clinic, it's called auditory habituation. And the key here is we play repeated beeps, beep, 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 4 at the same rate, and does your brain respond the same at the 4th beep, or the 2nd beep, or the 3rd beep as it did at the 1st. And what we see in Fragile X is that the brain is over-responding to sound. It doesn't sort of tamp down or tune out or tune down its response when it hears the same sound. And we think that this really relates to sensory overreactivity, and the brain just is too responsive. And can we correct this with treatment? And I think this is, again, conserved across animal and human studies. It's been used in a lot of trials at various centers, and it's a very consistent finding. All right, so this is my family. So I'm really happy to have them here. And we've been doing Fragile X conferences for over 10 years. They've been incredibly supportive. This is when I turned 40 in January, and they still opened up the Red Stadium so our whole research team could check it out and see Mr. Red and get a tour. So that was an incredible experience. And really, I think, you know, a testimony to our team and our relationships and the strength that we get working together. And then on the right, that's the Fragile X pumpkin, you know, with my kids. So, you know, we cut it out, we used the template, and we had a glowing X all night long. So that was a great experience. We've got a troll and a penguin there. All right, so now moving on to the results of our human study. So we study really age 15 and up. The age range of this sample, and I'm going to talk about the first half of the study. So we've got good data on 15 out of 30 individuals. Average age is about 26, range 15 to 41. IQ, and we have 2 ways of doing it. The bottom is a deviation score, really a methodology developed by David Hessel, who's here in the audience, that's going to really revolutionize how we evaluate cognition in Fragile X. It gives more variation, but clearly we have a cognitively impaired cohort. The final number is social communication. That's the variation of really autistic features in the group. So some folks with a lot of concerns for autism, some with a little. So there is really variation in the cohort or group. This is feasibility. I mean, can we even do it, right? So when we started doing EEG, I was somewhat skeptical, right? I mean, I've been seeing individuals with Fragile X for many years, and can they get the cap on? Can we get good data? And from left to right, this is the order in which we do the studies. We do resting first, then the chirp, then the habituation. We get almost 90% good data with resting at the beginning. Now, obviously, as you do things repeatedly, people get tired of it, right? They're bored. They don't want to do it. They don't like the cap. We drop to 75% with chirp, about 50% with habituation. But we're still getting good data on a large number of individuals. So I think we can consider this a feasible thing to do, in particular when we look at it's a broad population. So we are getting good EEG data on individuals that may be quite low functioning and have interfering behaviors. So I think that really increases the relevance of what we're able to do. The next point is, can we get the same EEG values today, in 2 weeks, in 4 weeks, in 6 weeks, before you even take a medication? Because if there's so much variation in what you're doing because of various conditions, you know, the air conditioner's turned on and it wasn't turned on, and, you know, somebody startled, or they were tired, or they started to like nod off while you were doing it, that would be a problem. But this slide, the take-home point is we can consistently measure these brain features across these modalities in a very consistent way in individuals. So by doing that, if we would detect change after dosing, we can attribute that more to the treatment than just the variation of life, that my EEG is one way on Wednesday and the next week on Thursday it's different. So this is really powerful, and we have to demonstrate this to use this in clinical trials, that it's consistent from day to day. All right, and the next look at baseline is, you know, who cares about EEG, right? Does it relate to the human condition? And what we see here, the gamma noise that we talked about, clearly relates to some human features. So what we see is that the baseline gamma abnormality, the excess, relates to clinical features of auditory attention and inhibitory control. So the more abnormal your brain gamma noise is, the more likely you have impaired auditory attention, and the more likely you make errors in computer-based testing that tests sort of your flexibility, or your ability to change sets, your ability to inhibit or stop when a pattern's changing, and those relate. So we have good data that the human condition, impairments in human Fragile X, are relating to this EEG work. So first of all, this is our resting state findings. So, you know, it's like a kid's book. Do these match or not, and what are the differences, right? So my daughter Julia is working a workbook right now, and I'm sure that's in there. You'll have these pictures, and what's the subtle difference and what's not. So we're looking at really the colored pictures. And is there a change post-treatment with a camprosate, Lovastatin, Minocycline, or placebo? And when you look down at that Minocycline bottom left, you can see on one side the red, which is signal intensity, is really going away. And that's our key finding that reaches statistical and what we think will be scientific significance. So the only significant drug treatment change we've seen in these first 15 individuals is in this right temporal region of the brain with minocycline treatment. We saw no overall whole brain effect, and we would consider this right temporal part of the brain associated with auditory cortex, or how you're processing and taking in sounds and language. And we're unsure exactly why it's on one side, you know, and I think what good science does is we create more questions than we get answers every time. We think it may be due to lateralization differences, or the idea that in individuals, it's been shown in autism, and we think in Fragile X, that there's an imbalance from side to side, and we may be restoring aspects of that balance. And on the chirp, you know, as awesome as the chirp sounds, and it's very interesting, we've not yet seen a significant human drug effect, okay? And again, it's a small sample. We're going to continue to enlarge the sample, but whether it's placebo or drug, we've not reached significance yet. But I think this is where it's key to increase the number of subjects and continue to probe the work, and that's what we're doing by getting to a total of 30, and we're at 15 with analyzed data now. And then this is the habituation effect, and this is the repeated sounds, and does the brain tamp down or tune out repeatedly? And we have a significant finding with minocycline that I'm going to talk about in detail on the bottom left. And the finding is the red is post-dose, the black is pre-dose. With the minocycline, the brain is able to attenuate better or get more used to the sound at that second beep. that we see down there with the blue arrow. And I'm going to talk about this now in more detail. So this habituation, or this sort of brain getting used to the sounds, increases following minocycline, and this is associated with increased auditory attention. So the wild bar graph there is just illustrative of that improvement at the second beep, correcting the brain's habituation, ability to tamp down its response. But the interesting part is, even in this small sample, the more habituation corrects, the more the brain is fixing its response, we see that auditory attention that we test with something called the Woodcock-Johnson improves more. So we are seeing a clear relationship between clinical improvement in change with a brain electrical function measure. And I think it's important to show these strong links, because otherwise, correct this in the brain on EEG, but how does it matter to the human condition? And I think, I want to point out what Rondi Hagerman, David Hessel, and others at UC Davis have done with minocycline in a small subsection of their minocycline trial, where they also saw some changes in habituation, in some changes in brain response to repeated sound. So I think we're beginning to recapitulate some of that groundwork work with only a single dose, which gives us the really interesting idea that we want to move towards, can a single dose profile in one day be used to predict longer-term treatment? So I think this is where the profiling and the prediction can be very important to improving human treatment trials. So this is an interesting finding that we continue to look at. Among those that received minocycline, improvements tracked together. So if your gamma power was improving, we saw improvement with the chirp. If your lower alpha, which is involved with cognition, was increasing in those receiving minocycline, we saw improvements with habituation, really linked up. And we saw this with minocycline, but not with the other drugs. If your chirp was improving, your habituation was improving, and they really correlate. So I think this is interesting, that we may need to really follow if these improvements tracking together correlate with clinical improvements tracking together with this drug versus other drugs. So, we still need to resolve this, but I think the sort of synchrony of positivity, that's like the niddering nabobs of negativism. I just made that up. But it's something we need to really ferret out and look into more. So this is a final comment on human research in the real world, right? Like, we cannot control people like you control fruit flies and rats and mice. It's the real world. So we went into doing this trying to do fasting studies. So the idea that individuals would, not be eating, they would maintain a fast, and we'd have predictable levels of medication in their system. And this gets back to that acamprosate quirk I mentioned. So Tmax is time to maximum drug level in your blood. And 6 to 7 hours would be great for our paradigm, where we wait 6 hours between EEG tests. But if you've been fed or you're eating, it goes out to 17 to 19 hours. So that's a big difference. And what we found is we can't keep folks fasting the whole day, right? I mean, we should have realized this, but, we're scientists and we just think that we can control conditions, right? But that's not true. So, in talking with our coordinators and getting feedback, people are eating. They're getting their Skyline chili, their La Rosas, they're having a good time. They enjoy that. That's part of the event. And so in practicality, we're dosing in a fed state. And what does that mean, in particular to acamprosate, given this really the difference between fasting and fed? And so what we found is we did PK, or blood concentration analysis, and acamprosate levels post-dose were really low. So at 1.5 hours, they were virtually non-detectable, and at 4 hours, right before the next EEG set, the levels were about 62 nanograms per milliliter. So just remember the 62 number and compare it to other blood level work we've done. So we've done acamprosate chronic dosing studies in Fragile X syndrome and in autism. And with somewhat similar dosing, our average levels are 8 times higher compared to what we're able to get with this single dose. And we give the largest single dose you could conceivably give, and we're getting 1/8 the level. So we're just not getting it through the system effectively. And if you compare that to what the published literature shows with single dosing, so sort of comparing apples and apples, at a dose half of the dose we give in adults, they see 3 to 4 times higher levels. So again, it's just in the fed state, not getting into the system appropriately. So what do we do? And how do we sort of correct ourselves? So the solution is, we talked to our scientific advisory group, is we've gotten rid of a camprosate from this model. Because you're not going to change the situation. We can't make people fast, and in a lot of ways, it doesn't seem fair or appropriate to continue scientific testing, giving them a drug with this setup, if we just know we're failing to get drug levels that we think may alter EEG and potentially have other improvements. So what have we done? Well, starting after the 15, we've replaced a camprosate with baclofen. And so a lot of folks have heard of R-baclofen or STX-209, a purified version. And this is racemic or mixed baclofen, and our advisors really weighed in on that. So we've changed what we've done. We've pivoted mid-study to really do better work and I think generate better data. Not that a camprosate has any problems per se, it just doesn't fit this paradigm. So we've moved on from that. And that is, I think, trying to continue to do good work, and you take the data as it comes. So limitations and future directions. Small sample size, 15 folks. We're going to get up to 30. We'll have 30 folks that have had placebo, Lovastatin, or Minocycline, and we'll have 15 of those will have baclofen data. We're bringing this approach into more drug studies. I'm doing work with AZD 7325. I'm giving that talk here in the session with Liz and Rondi on trial talks after this. And we're doing the same EEG paradigm. So we're taking all of our learnings into another molecule. Autism Speaks has recently supported a study in autism of minocycline. And it's interesting. It used to be that everything from autism gravitated over to Fragile X. Now the Fragile X world is driving the autism research world. And I think that's really a credit to what the many, many scientists at this meeting and others have done. But we're taking these learnings into a minocycline study. And then we're going to test other future drugs, both with single dose and after chronic dosing. So in that 1 day, what happens? And then what happens after weeks of treatment with your brain EEG. And then, finally, I want to end and spend, at least a little bit of time on, there are many thank yous in the work that we do. So first of all, the National Institutes of Health support this work. We cannot do it without the NIH. We really appreciate their support. I think this work is highly novel, linking animal and human study. And we couldn't do it without the dedication of the many families impacted by Fragile X who participate in our studies, who come to clinic. These are the folks we do the work for, and these are the folks that have really dedicated so much time and energy to these studies to try to improve outcomes. Our collaborators at Texas Southwestern, UC Riverside, Khalil Arena, and Devin, who's speaking after me, Mike Trinfalia from Fraxa, who really helped us deal with the acamprosate concern. And then these are overall thanks. So, you don't get to talk in front of the glaring lights a lot. So, you got to get out. This is the time to give your thanks. So, Chris McDougall is my mentor until we parted ways when he went to Harvard several years ago. He gave me the direction to enter the Fragile X field from autism while I was like a second-year resident or maybe even an intern. And it's been really incredible advice and really a great journey that I appreciate and will continue throughout my career. And then next, Lisbury Kravis and Rondi Hagerman. And I first saw them in action at St. Louis at the meeting in 2008 when they allowed me to sit on the medication Q&A session on Sunday. And it was kind of like a rock star show meets a Q&A meets a scientific session. And I learned that the connection with the families and bringing the science to families and to affected individuals is so important. And they really gave me examples by which I continue to strive to achieve. And then, And so our team, and I think in a lot of ways, one of the things I'm most proud of in the work at Cincinnati Children's Hospital and Indiana University before is the recruitment and training of great people that you work with. And I think you really want to be judged by the individuals that you're surrounded with more than even by yourself. So on the left side, we have our principal investigators in our group. So Ernie Petapati, who is doing really groundbreaking work with transcranial magnetic stimulation. He's doing great work with EEG analysis and really has an incredibly keen research mind. And then we have Rebecca Schaefer, who leads our psychology services and programming and treatment development that's not specific to drugs. And I really had the privilege to sit in a tremendous adolescent behavior session with her yesterday. And I think that's really a part of the program in research and clinic that is equally as important to what we do with medications. And then in the middle on the left is Logan Wink, who I appreciate came to today's talk because she has to cover when the rest of us are at a Fragile X meeting, right? I mean, that's a tall task. But Logan trained with me at Indiana University, came to Cincinnati when I did, and has really been a long-term collaborator. And she's doing great work now with pharmacokinetics and pharmacodynamics, so taking information about how much of a drug gets in your system and what effect is it having on your brain. And she's really building a tremendous and exciting career in autism using some of these modalities. And it really has been just an incredible pleasure to work with for what I feel like is like well over a dozen years. You almost quit counting years at some point. And then there I am glowing. And then the final person in the PI picture is Kelly Dominic, who joined our group and came out of the tremendous triple board program like Dr. Petapati at Cincinnati Children's in pediatrics and child psychiatry. And she's really doing groundbreaking work with infants with Fragile X syndrome and understanding their brain function and infants at risk for autism. And then the large group of glowing folks is our tremendous coordinator and research support team. And I really want to single out the individual on the left, Hillary Rossolo, who put together the event last night and is our lead coordinator and really is at the core of what we do. You know, I pride myself with never going over, but I'm a few minutes over, and this is the last of the pictures. But this is our broad clinical and research team through the years, right? So people come and go, and they go on to medical school, and they go to get doctoral training in psychology and neuroscience. And at the bottom right there, we're standing on the top of a parking garage during the eclipse, and I'm holding up my welder's glass. So that was a nice break in the day for everybody. And then we've got our trip to the Reds game. And so it's an ever-expanding and growing group, and there truly is no I in team. We couldn't do it without these folks that at the core are dedicated to clinical care and improving outcomes and using research as the ultimate tool to improve outcomes and help patients. And so I so greatly appreciate the team that we've built and the folks that I can work with. So thank you.
That's an impossible act to follow, but I'm really so pleased. Thank you so much, Dr. Craig, for inviting me and my research group, who's here at the table, to come share some of our work. One of the things that's really quite special that I want to highlight at the outset is being what I would term a translational neuroscientist. One doesn't really get to embody that word translation through disease as much as one would hope in many cases, but the direct collaboration with Dr. Erickson and his incredible group here at Cincinnati has enabled a true translational type of consortium where we're doing animal work, but we're using identical paradigms to as much as we can to what's used in humans. And we're really trying to do very, very similar things to what Craig was just describing, but in the mouse model. So I'd like to describe, and I'm going to kind of fly through a lot of data, but then we can come back to the end, maybe discuss it today. Some of the efforts that we've had recently in terms of trying to develop what we would call EEG translational biomarkers. In other words, what's different in mouse models of Fragile X in their EEG? Okay, I'm clicking. There we go. So, wait a second. Here we are. Okay, so the first thing, and of course this is exactly lifted from Craig's presentation, and I want to emphasize exactly that we're on the same page in terms of the EEG abnormalities, and specifically I want to focus on the gamma. There are changes in these other things such as Dr. Erickson described, such as alpha, but with the gamma, that's actually one of the main phenotypes we've seen in the animal model of Fragile X as well. And in particular, this question of auditory chirp. So I'll be focusing on gamma, resting gamma, and auditory chirp. So exactly the same parameters that Craig and his colleagues are looking at in the humans, in the patients, we're going to be looking at in the mouse model. Now, so what are the goals of our research? Well, stimulated, again, by the human data, and this is what reverse translation and forward translation, we want to do both. Develop measures of the sensory circuit dysfunction in the Fragile X knockout mouse model, which is the so-called Fmr1 knockout, using neurophysiological methods, particularly EEG, and then looking at this resting EEG and also sensory stimulation, auditory stimulation evoked EEG, and seeing whether that leads to predictable phenotypes that we can then evaluate. And then, in more particular for the purposes of this talk, number one, do these Fragile X knockout mice have increased resting gamma power? And do they have reduced phase locking to auditory chirp? I'm not going to go through the definitions of that because Craig just described it, but it's exactly the same type of question in the mouse. And then, given that, do drugs, particular drugs in a single dose model, very much paralleling what Craig described, have effects on EEG parameters? And we directly collaborating on these questions. So the first question of, why would we do this? Well, first of all, as Craig described, compared to marble burying and things of that nature, EEG is a more sort of what one might call objective physiological criterion in terms of an outcome measure, and it's something that we can quantify. And the other thing is we're doing these experiments with the mice awake and freely moving. And for those who don't do animal research in the room, I want to assure you that we do absolutely everything. These animals are very well cared for and they're not experiencing pain. And I'll show you an example later of an animal moving around a cage connected to the EEG, but we take good care of them. And then the fact that they're not under anesthesia. We started these studies a few years back with Johnny Lovelace, as I'll describe, under light anesthesia, but that affects the EEG. Now we're doing all of these with awake, freely moving mice, which avoids the confounds of anesthesia. And then, importantly, the data collection, such as Ernie Pedapati's EEG pipeline and other things we're now developing to do in parallel with both mouse and human data, so we can really try to compare these directly. I want to start with Johnny's data, which has been really quite remarkable over the past few years. He's had several groundbreaking papers on this, where basically he pioneered this so-called, what I'm calling a two-channel EEG method, but where mice could be implanted with electrodes over the frontal cortex and over the auditory cortex with an occipital reference. And then the basic paradigm, and this is not going to differ throughout so I'll kind of describe it now and then kind of flip through it later. The basic paradigm is the mice are implanted with the electrodes. They recover after the procedure. And then they are brought to a recording chamber, which is a sound-insulated chamber where sound stimuli can be carefully presented. But then there's a baseline EEG, which is shown there as EEG testing baseline. And then we give them various types of auditory stimuli, such as the auditory chirp stimuli. And these are done in what are called C57 Black 6 mouse background and wild type and knockout mice. Wild type meaning they don't have the Fragile X knockout, and knockout mice are compared directly in terms of the EEG. And the finding, which is actually quite similar in many ways to the human data, and I want to focus on the red part, which is the gamma range EEG. You see we have all of these different EEGs, delta, theta, alpha, beta, gamma, as Craig described. The gamma is the higher frequency range. In this particular figure, it's 30 to 55, although we've also looked at higher gamma. And basically what you see, and it's quantified it on the right, what this is a ratiometric it's a ratio between the knockout and wild type gamma power. So if at 1.0, and I hope you can see that black line which goes across at 1.0, that would mean the knockout and wild type mice are exactly pretty much the same in terms of that power. But you see, especially into the red range, that there's this rise in the gamma power, and that means that as a ratio, the knockouts are having more gamma power than the wild types. So this is how the data that I'm going to describe today are being presented. And that's something that was true in both the auditory cortex, or AC, which is the top one, and the frontal cortex, or FC. And of course, with Johnny's information, we were able to look at both the auditory and frontal cortex in this way. And then we, of course, wanted to evaluate The chirp stimulus, and of course this is what the one that in humans, thanks to the data from our colleagues and collaborators, showed that there's a reduction in synchronization to the auditory chirp stimulus. This turns out it's a down chirp. You can do it a down chirp or an up chirp, and I'll show you up chirp data as well later. But you can see here in the middle panel that the knockout compared to the wild type on the left has a lighter color, meaning not as bright. That's a color-coded correlation of what's called a phase locking factor. How well is the brain locking to the stimulus of the auditory chirp? And you can see that over those 2 seconds of the chirp, which is a broadband noise stimulus that's presented as described on the bottom, the knockout mouse, both in the frontal cortex, which is the bottom actually, and the auditory cortex, which is the top here, does not respond as well in terms of phase locking to the chirp. So this is very much along the lines of what had been seen in the patients with Fragile X syndrome. So we have now a mouse model where do Fragile X knockout mice like humans, do they have increased resting gamma power? Yes, now with this we can show that they have that. And do they have reduced phase locking to the auditory chirp stimuli? And yes, indeed, that's also the case. So with these earlier data, with these initial data, we now know that we can use this Fragile X knockout mouse to sort of model these aspects of circuit abnormalities or circuit differences in the brain, both in the frontal and auditory cortices of the brain, that there's increased gamma power and there's also this reduced phase locking. Our next goal, and that was published recently by Johnny in 2018, our next goal was to actually develop this more widespread throughout the brain. We want to look at many different brain areas and targets and be able to do that. To make a long story short, we wanted to get multi-channel EEG data. Beyond just the initial 2 electrode data, we wanted to actually be able to model this better. One thing, as Craig showed you with the humans, is they have this cap with, of course, many, many electrodes on it. What's interesting is when we started this work, there was no equivalent way to do this in mice. And so that's actually something that we've been working quite a lot on, and Carrie Jonak, who I'll show her picture in a moment, in the lab has been instrumental in setting this up and really developing this to basically do what's called multi-electrode arrays, or MEAs. And the idea, of course, is that the humans have 128 leads. In the initial mouse data, we had 2. We were getting great data with that, but as Johnny showed, but we wanted to see if we could do something to develop MEA in the mice. And the concept, of course, is adding all these more electrodes would give us more spatial information throughout the brain and potentially more sampling and reveal new findings. The hardware for this is as described. There's the mouse is connected to something called a head stage. The details of that technically are that allows digitization of the signal at the head of the animal, which minimizes things like noise and movement artifact. And then there's something called a commutator, which you'll see as the mouse is moving around. We don't want movement artifacts in the EEG. And those EEG people in the audience know exactly why that's important. And then it basically enables free motion of the mouse during the recording period. We have then, in terms of what actually contacts the surface of the head, something called a flexible surface microelectrode array. And we kind of experimented with a variety of different ways to try to implement this in mice. And Carrie, whose picture is in the upper right corner there, has really done an amazing job on this. And we ultimately came down to using the system that's in the bottom there, which is a 30-channel array. And each of those little dots there is basically a small electrode that's connected by these microwires to basically that ribbon at the back, which Carrie then can implant very successfully in these mice and keep it on for long periods of time. The other thing is it's implanted in the exact same location, and because we can see the sutures this is on the surface of the mouse skull, we can see actually how to basically put it on the mouse the exact same way between mice so we can compare spatially as well. That took quite a lot of work, but it's working well now. And then what you end up with is a mouse that is doing well, is moving around the cage, and this is an area, you can get baseline EEG, you can do the auditory chirp stimuli, etc. So very similar to Johnny's protocol, the mice are implanted now with the multi-electrode array, and then they recover for, in this case, a couple days. and they're doing well, and they go into the cage, and then or into that recording chamber. And then basically you get the resting EEG, and then we get the chirp trains and things like that. So we average actually a lot of this there's actually 300 chains, so it's kind of averaged over many, many trials, just the details of that. And what I'm going to show you here, actually everything from here on is unpublished data, so this could be considered preliminary, but we're excited to share it with you today. This is actually from 6 wild type and knockout mice. And one thing that we noticed, that Carrie noticed early on, is that actually just putting down these multi-electrode arrays, we end up getting very stable EEG, and actually all 30 channels which are shown there, you can then all kind of plot together. It's all these little squiggles. But if you get an idea, even just looking at it grossly like this, the wild type and knockout EEG just looks different. And how many people could actually say that it looks different, even if they've never done EEG in their career before? Oh, that's encouraging. Thank you. Makes me feel better that I'm not just, because I asked my son, who's 7 years old, and he's like, yeah, it's a little squigglier on the right. But that's exactly right. And what that is that high frequency activity in the EEG that actually is higher, and that's the gamma. We quantify that. Now, I don't expect you to read these axes because it's 30 channels, but just to give you the idea that now we can analyze, as compared to the 2-channel data, now we can analyze all those little spots on the surface of the skull differently. I recognize we're not on the brain surface, we're on the skull surface. The reason we did that is we don't want to actually damage the brain at all during this. We want a high fidelity recording from the skull surface, which is basically what we're saying is equivalent in a very basic way to the human cap. Obviously it's different. It's 30 channels in a mouse versus 128 in the human, but we're getting closer to try to get spatial data in the mouse model. And that's the rationale for this. But of course, to try to analyze these data, because you end up with 30 channels, what are you going to do with all that? We've now grouped them, at least just provisionally, into these 6 brain regions: left and right frontal, which would include the boxes there in terms of all those electrodes. And we can go back and analyze each electrode individually, but this is an idea. Then we temporal regions, like Craig said, which are on the side of the head, and medial regions, right and left. And then, of course, also related to this interesting right-left difference that Craig was mentioning, we can look at right-left differences in the mouse as well. This shows the finding that, similar to Johnny's findings before, that with the multi-electrode arrays, we're getting increased gamma in the red, and there's also some changes in lower frequencies. But this is the frontal region, and then we can analyze the medial region, which doesn't seem to be as changed here, and the temporal region. So we can kind of spatially look at gamma through the brain, and there's significant increases in gamma as described before. Then you can actually collapse all the electrodes down into one, average, and you can see that indeed the knockout over wild type ratio is above 1, which is again the same way we're showing these data. In other words, the red mountains that are above the black line indicates that there's increased gamma in the knockout. So now we have this filled in here, where not only with the 2-electrode system, but with this multi-electrode array, we can now see that we see increased resting gamma power similar to the humans. Now the question is, what about the phase locking to the chirp? So we can do that same protocol with the 30 channels. And that's what Carrie has done here. And this, again, is now that same type of up chirp that you heard from Craig before. And if you can look at all these little lines here, you'll see that the strength, basically the color coding, more red in the wild type compared to the knockout at each channel. But then, of course, we can also bring it down into these 6 regions, such as left and right frontal, left and right medial regions, left and right temporal regions. And what you notice actually is a couple interesting things here. One is that the wild types, and keep in mind this is 6 and 6 each, but the wild types are stronger in terms of the phase locking factor to the chirp. And just to remind you, that's this 2-second stimulus of increasing frequency, where the brain is trying to lock to that stimulus. And the wild types are stronger than the knockouts. But you also see, and this relates to some interesting discussions we're having with Craig and colleagues, the right side seems to be stronger than the left. So there's this interesting potential change between right and left, and we're still in the process of analyzing that more in detail. Of course, the brain is lateralized, and it may be a real finding, so we need to statistically analyze that. If you collapse everything down into one electrode, you get a huge difference here in between wild type and knockout in terms of the fidelity of this phase locking to the chirp stimulus. So we're now able to really see this with just a few mice in each group, which is encouraging to us because we can also use this potentially for drugs. Drug effects. So yes, we also have reduced phase locking to the auditory chirp stimuli with the multi-electrode array system. So the next step, or the second big thing that we've tried to start with, and again, this is all very new, but there's a very fresh data over the past just 2 or 3 months, basically. Do drugs, such as the ones that our clinical colleagues are testing in Craig's studies, do they have effects on EEG parameters? The same exact drug. Does it have an effect in the mouse in terms of these EEG outcome parameters? So we started with Minocycline, which is this is a single dose design, and this is, obviously it's not the same dose as human, but in the mouse it's 50 milligram per kilogram. That's based upon other studies. This is given intraperitoneal, meaning basically into the belly of the mouse. And this is 1 hour later we do, so actually we get a pre and a post. So in these studies we of course want to get a resting EEG before drug, and a chirp, and then of course the minocycline is given, and then we do the same thing an hour later. And keep in mind, because I want to show you, these are very preliminary data, it's just N of 3 per group, so really we need to do more of this, but just to give you an idea of the type of things we're doing stimulated by our human colleagues. And this just shows the resting, which is knockout over wild type, and again the knockout has more gamma to start with. What's interesting is there's wild type over post over pre, so now this is a comparison of what does the drug do to the wild type, and it doesn't have a huge effect here on the EEG in terms of big changes. There's not a huge effect here on the knockout either in terms of resting or post versus pre. I'll show you the other drugs that have more an effect. You'll be able to compare. And then the wild type pre and post chirp, we're seeing really not a lot of significant differences there. Again, just 3 per group. And this relates directly to the fact that, Craig was saying that the minocycline, they're seeing some slight effects, but not dramatic effects, and not effects on chirp on the acute dosing. And I'll come back to that at the end as a discussion point. What's interesting here in the knockout, however, there seems to be slight improvement if you look very carefully. For example, in the right medial, the right and the temporal, there's some potential improvements, slight though they may be, in the chirp with the knockout with minocycline. More impressive though, and I'll come back to that again in a moment, are some of the other drugs that we've looked at. So here's a Lovastatin dosing of 30 milligram per kilogram, and again, pre and post, 1 hour after, just giving it to the mouse. And it doesn't do a lot to the baseline in the wild type, in the knockout or wild type, in terms of actually the amount of effect on gamma, although there is, just go back one. In this case, the Lovastatin does appear to reduce gamma in the knockout mice. So if you see the red going down below the black line, so you can see that there's a reduction in gamma by the Lovastatin even 1 hour afterwards. The phase locking factor to the chirp, basically the chirp appears to improve from wild type pre to wild type post, and also from knockout pre to knockout post. In other words, the drug itself is improving the ability of the brain to respond to the chirp in this paradigm. I will skip that slide just for now. Racemic baclofen, this is the newest drug that Craig and his colleagues are testing as he described just in the last talk. We've now given this based upon a nice study from 2014 on dosing this 2.5 milligram per kilogram IP in the mouse, 3 and 3, again, just getting started with these studies. And it doesn't appear to do a whole lot to excuse me, these are knockout pre to not wild type pre, meaning there's not a lot of, there's already a baseline difference in gamma in knockout versus wild type. This is before drug. After drug, it's interesting, so wild type post over pre, you see the red is below the black line. What that indicates is that the gamma power has been reduced by the drug 1 hour after, and that's quite, that appears to be quite significant. And he was sort of significant in a statistical sense, but it's a large effect. And similarly, in the knockout itself, it seems to reduce the gamma. So then what does it do to the chirp? Well, it's quite interesting, and racemic baclofen appears to significantly improve the phase locking to the chirp in the wild type, and then also in the knockout. This is probably the biggest effect we've seen so far in terms of the amount of improvement in the chirp phase locking. And if you actually compare the pre-treated wild, meaning untreated wild type, to the knockout after treatment, it's almost like it rescues or tends to make it much more like the wild type. We recognize that these are sort of just early data, but it's exciting to see that, for example, in this case, that there's quite a lot of improvement in the chirp stimulus and also a reduction in gamma. I want to summarize just where we are so far, and of course there's a lot more to do. This is early data, but we've developed and applied multi-electrode arrays to the knockout Fragile X mice in vivo for the first time. And we've shown feasibility in obtaining resting EEG and stimulus-evoked EEG in awake behaving mice. And we found, and this is very, very congruent with the human data, there's enhanced resting state gamma power, and there's a decreased ability to phase lock the EEG to chirp stimuli. And there are very similar results, although differences here and there, between the 2 electrode recording system and the multi-electrode arrays. And then in terms of these early data on single dose trial-induced drug changes in EEG parameters, it appears to affect both, in certain cases, both resting EEG and phase locking. And specifically, I want to kind of describe the following. So what are the drug effects so far that we see on gamma power and chirp? Well, first of all, and this relates directly to what Craig said last talk, which is if the drug tends to reduce gamma power, it also seems to improve chirp. And it may also lead to compensatory changes in powers at the lower frequencies, which we're also in the process of analyzing. But taken together, our drug data suggests this. So for example, acute 1-hour minocycline did not change gamma power significantly, but similarly minimal effect on chirp phase locking. But the acute Lovastatin, in contrast, did reduce gamma power and improve chirp. And the same thing with racemic baclofen in both wild type and knockout mice. I want to mention then is something more like chronic treatment with minocycline, because it's an enzyme inhibitor, a matrix metalloproteinase 9 inhibitor, and it may have a slower time course, may have an effect at longer time points and not at the acute stage. And that would be something that we could use the mouse model to potentially predict the single dose in the human trials versus longer dosing. And Johnny Lovelace has specifically analyzed this with the 2 electrode system, and I'd strongly encourage those of you interested in this to attend his talk tomorrow, because he's going to go over these data in more detail. Well, in summary, these data have all come out of this consortium, and I'm really pleased and honored to be involved with this NIH Collaborative Center for Research Fragile X. My UC Riverside colleagues, Khalil and Irina, co-PIs in this work. But the work I've done, that's been done, that I've described today, is really from Johnny and Carrie, so I thank them very much for their incredible work in this region. And thanks for your attention. I'd be very, very happy to take some questions. Thank you.